Hello and welcome to my presentation on antimicrobial and immunomodulatory activities of self-assembling peptides and peptide nanoparticles. My name is Nauman Nazeer, and I am a PhD candidate in molecular and macromolecular science at the University of Prince Edward Island. My research is being conducted under the supervision of Dr. Maria Ahmed. To provide a quick outline of this oral presentation, I'll first go through a short introduction to this topic, followed by a detailed discussion of the major findings of my research, before ending with a quick summary of this research. To introduce some background behind my research, the widespread use of antibiotics in the environment has led to an increase in antimicrobial resistance among many common pathogens. This is soon to become a serious challenge to public health in the near future as these antimicrobial resistant strains proliferate in the environment and soon render many commonly used antibiotics completely ineffective. This necessitates the need to develop new classes of antibiotic drugs. One major class of antimicrobials that are being widely studied to replace conventional antibiotics are antimicrobial peptides. These peptides are part of the innate immune response system of all living organisms, and they are generally cationic in nature, containing residues such as lysine and arginine. They show strong interactions with the phospholipid bilayers of cells and are highly selective towards prokaryotic bacterial cell membranes due to the presence of more negatively charged acidic phospholipids. However, they are much less selective towards eukaryotic cell membranes due to the presence of more Zwitter ionic phospholipids as well as cholesterols. In order to develop our peptide library, we looked at chicken angiogenin 4 or ChANG4. This is an antimicrobial protein. Some of the important regions of this protein are highlighted here, and these are active sites which we looked at to derive new antimicrobial peptides. From the cell binding segment of this protein, we derived a series of 4 antimicrobial peptides. And from a subdomain of ChANG4, we derived a 5th peptide. And out of the 5 peptides, 3 of them are cyclic peptides that have cysteine at both ends and are disulfide linked. These peptides were synthesized by solid phase peptide synthesis, and their identities were confirmed and characterized by mass spec, and all of these peptides have a net positive charge of +3. The antibacterial activity of these peptides was studied against a series of both gram-negative and gram-positive bacteria, and it was found that only 2 of these peptides had any significant antibacterial activity with a 50% inhibitory concentration or IC50 below 100 micromolars. And out of these peptides, the one highlighted here showed a comparative antibacterial activity to that of a commercial antibiotic, ampicillin. The antimicrobial peptides that we developed were found to self-assemble from random coils to anti-parallel beta sheets, which further self-assembled into disulfide-linked nanospheres or DSNs. The use of polysaccharide-based carriers was found to stabilize the self-assembly of these nanoparticles to give more stable DSNs that showed improved antibacterial activity, improved membrane permeabilization, improved bacterial flocculation capability, and improved immunomodulatory activity. In order to produce stable DSNs, two polysaccharides were mainly studied. These were fucoidone, or Fc, and β-cyclodextrin, or beta-CD. Fucoidone is an anionic polysaccharide derived from brown seaweed that could form electrostatic complexes with our cationic peptides, while beta-cyclodextrin is a cyclic polysaccharide derived from starch that has a hydrophobic cavity that can incorporate hydrophobic molecules, and therefore it is commonly used as a drug delivery carrier. Transmission electron microscopy, or TEM, was used to observe these nanoparticles, and it was found that the DSNs were compact, having a size of around 15 nanometers. The antibacterial efficacy of these nanoparticles was studied against a range of gram-negative and gram-positive bacteria, and it was found that the beta-CD DSNs were the most effective, having the lowest IC50, lower even than that of ampicillin. The bacterial flocculation capability of these peptides was also studied, and it was found that these peptides caused the aggregation and flocculation of bacteria in solution, and this ability was maintained even by the nanoparticles of these peptides. By looking at fluorescence imaging and live dead staining, it was found that the flocculates indeed consisted of dead cells and not living cells. The same result was found to be true for both gram-negative bacteria such as E. coli as well as gram-positive bacteria such as B. subtilis. To study the effectiveness of these peptides and their nanoparticles at disrupting the cell membranes of bacteria, a membrane permeability assay was conducted in which bacteria in suspension were treated with these peptides alongside with a hydrophobic fluorescent dye, carboxyfluorescein. This dye would not permeate the cell membranes of living bacterial cells where the membranes were intact. However, they could pass through ruptured membranes. It was found that the uptake of carboxyfluorescein was increased in the bacteria that were treated with beta-CD DSNs, much more so than the other DSN nanoparticles, and the uptake of carboxyfluorescein was overall increased compared to a control in the case of all peptides and peptide nanoparticles. Physiological stability of the peptides and their nanoparticles was also studied in both PBS at a pH of 7.4 as well as a solution of 10% serum proteins. It was found that DSNs as well as Fc DSNs were very physiologically unstable. They were found to aggregate in both PBS as well as in 10% serum solution. The beta-CD DSNs, on the other hand, were found to have much greater serum stability. Beta-CD DSNs were also found to maintain their antibacterial activity in serum against E. coli, whereas other DSNs were found to have a statistically significant loss in their antibacterial activity. Cellular uptake of DSNs, FCDSNs, and beta-CD-DSNs were also studied in both gram-negative E. coli and gram-positive B. subtilis, and it was found that all of the DSNs were steadily uptaken by these bacteria, with beta-CD-DSNs showing the greatest amount of uptake. Fluorescently labeled peptides were also used to study uptake in Caco2 adenocarcinoma cells by flow cytometry, and it was found that beta-CD-DSNs showed much greater uptake, more than twice the amount of uptake compared to other DSN samples. It was also found that the DSN samples were completely non-toxic towards DF1 fibroblasts and Caco2 cells, indicating them to be non-toxic to host cells. The cytotoxicity studies were done at a concentration of 100 micromolar for all DSN samples. It was also found that these DSNs were relatively non-hemolytic against red blood cells, with beta-CD DSNs showing the lowest rate of hemolytic activity. It was also found that DSNs were very effective at neutralizing the effects of bacterial toxins such as LPS or lipopolysaccharide and LTA or lipoteichoic acid, which are produced by gram-negative and gram-positive bacteria respectively. Beta-CD DSNs were again found to be the most effective. Interestingly, however, it was also found that cyclodextrin on its own was also a very effective agent to neutralize the effects of these bacterial toxins. And finally, the anti-inflammatory ability of these peptides and their nanoparticles was studied. It was found that all DSN samples were able to reduce the production of pro-inflammatory cytokines in macrophage cells that were challenged with LPS. They were able to reduce the production of cytokines such as interleukin-1 beta, interleukin-6, interleukin-10, and interleukin-12, and it was found that beta-CD DSNs were the most effective at reducing the production of these cytokines. So, in summary, we developed self-assembling cyclic peptides that formed anti-parallel beta turns and beta sheets and further organized into DSNs. They displayed antibacterial and bacterial flocculating efficacies, and although they were physiologically unstable in the presence of serum, they could be made physiologically stable by complexing them with fucoidone and beta-cyclodextrin. The beta-cyclodextrin DSNs also displayed improved antibacterial activity and improved immunomodulatory activity with reduced toxicity to host cells. This is my list of references for any additional information provided in this oral presentation. And finally, I would like to thank and acknowledge my supervisor, Dr. Maria Ahmed, as well as my committee members and co-authors in this research project. I would also like to thank my colleagues from the Ahmed Group at the University of Prince Edward Island, and I would like to thank technicians at the University of Prince Edward Island, as well as staff from the National Research Council of Canada and from Dalhousie University, who were instrumental in providing data to determine the secondary structure of these peptides. And finally, I would like to thank the various funding agencies that have made my research possible, and I would like to thank CCCE 2021 for providing me the platform for presenting my research. Thank you.